This protocol has a high impact in the field of drug discovery and development, especially in the development of novel first-in-class drugs with minimal side effects. The main advantage of this technique is that it can be applied to any GPCR system and used to obtain accurate information about receptor pharmacology in real-time living systems.
The implications of this technique are closely related to new therapies, particularly where beta-arrestin are relevant. These include opioid receptors in pain and dopamine receptors in schizophrenia. This method can be useful in many medical scientific research areas, from neurology to cardiopulmonary diseases.
Visual demonstration of our method is important because it allows for a detailed presentation of the critical steps in the protocol, which would not be possible with a conventional publication.
Begin by designing primers to introduce genes of interest to the p-bit vectors. Due to the in-frame stop codon that divides the multi-cloning site, select at least one of the 3 sites as one of the 2 unique restriction enzymes needed for directional cloning. Refer to the manuscript for nucleotide sequences to encode the linker residues and incorporate them into the primers. For the p-bit 1.1c and p-bit 2.1c, make sure that the forward primer contains an ATG codon and a potent Kozak consensus sequence. For the p-bit 1.1n and p-bit 2.1n, make sure that the reverse primer contains a stop codon.